Okay, this video all about muscles, how they move, how different kinds of muscles move, how the little subunits of muscles move. A lot of really interesting stuff, a lot of very intricate stuff today. But alas, we press on and move forward. Very interesting stuff. Kind of hard to learn though. But I'm gonna start out with the sarcomere. Because this is the subunit, the contractile subunit. That's what I'm gonna call it today. If we actually no, this is a muscle. Duh. And then if we zoomed in on this muscle, we would see that it's made up of a bunch of different fibers like this. And then if we zoomed up even further onto each of those fibers, it is one fiber that has all of these little contractile subunits, like I mentioned earlier, that are called sarcomeres. So sarcomere is just one part of one muscle fiber that's part of a muscle unit or a motor unit. And then if we zoomed in even further on this guy, then we get something that looks a little bit like this. We have different types of little filaments, different types of units, muscle fibers. They're very, very specialized in what they do, and very, very good at what they do. These little lines up here, these are called nebulin. These little lines. Hey,
are you making a video?
Yes, I am making a video.
So sorry.
These lines, we call titin. So titin, and then this is nebulin. Are these lines? And then if we even wanted to zoom in even further, we're gonna go to just, if I added all the little things on here and then zoomed in, it'd make a little bit more sense. So if we just zoom into this guy, we would see that we have something that looks a little bit like, here's the nebulin, and then we have something called actin, little actin filaments. This is just the shorthand of how I'm going to draw them. How they actually look is we have things called F-actin and G-actin. I know the F-actin is filamentous actin, but basically, so this is like one actin, and then they're like in a chain like this. And they have these little myosin binding sites, which is very important for later. So please keep that in mind. Actin binding sites. And then we have what we call tropomyosin, which is like this just long curvy little line guy that just goes all around them, which binds to the myosin binding sites. And you may be asking yourself right now, wait, aren't they called myosin binding sites? But I guess tropomyosin kind of has myosin in it. Fret not, that will be solved later. We also have something bound to the tropomyosin, which is called troponin. They look like, we called them little Mickey Mouses in class, but they look like that. So like this is, feature this is that. So what we need to do in order to contract muscle is get the actin free of the tropomyosin and the troponin. And so what we need to do is we need to add calcium, because these guys have three different subunits. We have the C, we have the I, we have the T. The I binds to the actin, and then the T binds to the tropomyosin, and then the C is what's going to be to bind to calcium. So we have high infinity areas and low infinity areas. So when there's enough calcium in the environment in order to bind to the low binding sites, that's when these get released off of this, and it brings, since it's bound to tropomyosin right here, it takes tropomyosin off. So when I'm gonna erase this arrow really quick. So when we add calcium to this, then these guys go away, which then in turn makes it so these actin binding sites are now open to be bound to get this myosin. Break. Okay. Next on our list is, but Paige, how did the calcium get into the environment in the first place? Good thing I have an answer for you. I'm gonna switch to green because why not? So something about muscles is when you hear muscle, you just gotta think calcium, because calcium actually literally does everything. Very, very important. I mean, other things obviously play a role, but it would all be for naught if we did not have calcium. So we're just gonna draw a little muscle guy. This is going to represent a little neuron. This is like the terminus, and then this is going to be the edge of our muscle. And then this is something we call a T-tubule. I don't know if you can read that, but it says T-tubule. And so what we're going to have here is we're going to have little sodium guys. I don't know if it's sodium, but little channels that allow the positive charges to come in. Charge. There's calcium channels up here. Not calcium. Sodium. Channels up there. No, it is calcium. Oh well. Calcium channels up here. So what happens is that these guys are basically full of little vesicles, and vesicles are the little taxi guys of biology. And these vesicles carry neurotransmitters, and then they release neurotransmitters into this area. Then it can enter into the cell. So the cell of a muscle contains what we call the smooth endoplasmic reticulum, the SER, or for shorthand, it might just be referred to as the sarcoplasmic reticulum, but it's really called the sarcoplasmic endoplasmic reticulum, I think. But smooth ER, or SR. I will probably reference it as SR. So this guy, just chock full of calcium. That's its job, is to hold calcium. Calcium, calcium, calcium. And it's really good at its job. However, we have these little proteins, little receptors. I guess I don't know what they're actually called, but they are attached to the side of the T-tubule as well as the side of the sarcoplasmic reticulum, or the SR. So these two guys are called DHP, and then these two guys are called RYR. And then I'm gonna move that a little bit because I need to draw under it. RYR. And then this guy also has something called calsequestrin attached to it. I'm not gonna write it out because it's a long word, but it basically just has a huge negative charge, so it attracts the positive charges of the calcium. That will be explained in a second. Okay, so this is basically how your little muscle cell is set up, and then here's where the action potential happens. The positive charge, there's voltage gated channels all along this, but when the Bourn, where the sphere of influence reaches these guys, they open up, which is super, super important, because this, like, your muscle is depending on your sarcoplasmic reticulum to supply pretty much all of the calcium you're going to use. Like, you have some in the extracellular fluid, but that's used for a different thing, kind of. But we're really focusing on trying to get the calcium out of the sarcoplasmic reticulum. So what happens when the action potential reaches these guys? They're going to go through a conformational change. So they're basically just going to change shape, is what that means. And they're just going to open up and leave these gaps. Wow, I cannot draw squares. So then calcium can leave and go into the cell and onto those sarcomeres and onto the troponin, which releases it, as well as taking the tropomyosin. So it just like has this cascade of, it's very important. So we like that this happens because we really need calcium. We also have these pumps right here on the sarcoplasmic reticulum, the SR, and they're called circopumps. Smooth endoplasmic reticulum calcium circle pumps. And they just make sure that there is a reuptake of calcium just to keep replenishing this store. And so like I was saying before, this calsequestrin has a super, super negative charge. And so when the RYR opens, all of the calcium is already like right there and ready to go. And then it can leave and go into the cell. There is a little bit that ends up in the T-tubule, and there is a little bit that comes through here, but that's not really the problem, or it doesn't really do anything, so we're going to ignore that for now. Okay. Next, we're gonna head back to the sarcomere now. Sarcomere. I'm gonna stick with green because I like green. All right. I'm just gonna draw another sarcomere. We have the nebulin. We have the titin. And then nebulin. So on here is where myosin hangs out. We love myosin. Myosin does a lot. And so I'm going to just add calcium into this environment. So then the actin is up and available. So that means that those actin binding sites are open and are able to actually bind to actin. And so these guys want to go there. So that is what we're going to talk about, is when they link together and how they actually pull the sarcomere together, like the Z discs together, and create like the shortening of the muscle, like the contraction of the muscle. And so if this is the Z disc, this is I'm just going to call that actin. That line is actin, and then here's the head of myosin, and then the tail. So this guy, step number 1, this guy, he is in the cocked position, as well as he has a little phosphorus on him. Right? Yes, he has a little phosphorus on him, and he is just chilling, and so we need to do a couple of things in order for it to actually attach to actin. So what we're going to do is we're going to, again, add calcium into the environment. Calcium! So this is going to, I don't actually know what calcium does here. However, the next step is that the head binds to the myosin, but still has that phosphorus attached to it, which is kind of a problem. It's just blocking it from getting into the cocked position, which is what we want. The cocked position is actually what's going to move the actin and then move the sarcomere together. And so then the next step is that we're going to kick off that phosphate, say bye. We're going to kick off, there's also an ADP on here, adenosine diphosphate. So we're going to kick off ADP, DP, and then, we're going to go to what we call the power stroke step. So the myosin head is attached to the actin. However, it's going to go through another conformational change, like the RYR and the DHP did. It's going to change shape, and it's becoming the cocked position. So it's going to move this that away. Next, but that is literally so small because it's just the subunit of a muscle. So moving this much of the muscle isn't going to do a ton. So it needs to be able to release, rebind, and go back into the uncocked position in order to move over and over and over again, which causes muscle contraction. So we're going to add ATP because in order for it to, I'm going to erase this a little bit. In order for the myosin to release from the actin, it needs ATP. And so we're going to draw the Z disc again, myosin, and then I'm going to add an ATP to this because it needs to release from the myosin. And then here it goes through what we call, I'm not going to write it down, but hydrolysis. And then the ATP comes off, well, not really comes off, but one of the phosphates comes off of the ATP, binds here, and then we have ADP and phosphate on a cocked position myosin head. And the cycle starts all over again. So this is called power stroke. Yeah, and then that's how your muscles move. It basically just goes around and around and around and does this over and over again until your brain is like, yo, you can stop that. And then they're like, okay. So then they stop, and then your muscles relax, and you go back to your original position. So that's how that works. Going back to red. All right. Next, I'm going to talk about. I don't like how I start with that. Let's talk about energy sources that are available to your cell and the circuitry used for energy. There are basically, from what I understand, 4 different types. There could be more, but I'm just going to talk about 4 of them. The first one is called cyclic ATP. This guy does a couple of things. It basically is just like a pool of energy that your cell can use. Like, it's just there, it's ready, it's not like assigned to a job or anything. It's just waiting to be used, and so your body's like, yo, I'm gonna use that. However, it's very, very short-lived, and it only gives you like a second of movement. And so it's really good because there's a lot of it, but it isn't sustainable in the long-term run, and long-term, whatever. But your fast twitch muscles use a lot of this. So like your eyes that are always like are always using this stuff because they don't need more than a second of movement, and so this is fine to use. But like your quads aren't going to use cyclic ATP because it's just, you don't need to do that. You just use a lot of it and it wouldn't be very beneficial. The next one is called creatine phosphate. And what this guy does is it takes a phosphate and an ADP and then makes ATP. This also is another short-lived one. It's a little bit faster. I think it's like 10 seconds. Like 10 seconds of movement. So it's a little bit more and available to use, but still, again, not very good for like the long term of moving muscles. So this will still be like your fast twitch and not really large muscles, I guess. Small ones maybe like in your hands. And then the next one is called glycolysis. Everyone knows glycolysis. Everyone loves glycolysis. Very complicated. However, it's basically just the breakdown of glucose. But there is a caveat that all 3 of these are anaerobic. So this isn't cell metabolism. This is just the anaerobic portion of it. This guy just breaks down glucose into ATP. It actually provides a ton of energy. Not as much as if you went through the electron transport chain, but still a lot. And then this gives you like a minute of movement, as opposed to like the 1 second or the 10-second one. So it's longer than the first 2, but still not good for long-term use. However, there is an aerobic one, which is oxidative respiration. So your cell's metabolism, it's just, again, breaking down glucose but using oxygen, and so it just gives out a ton of ATP. If I could remember the Krebs cycle and stuff, I would write it out, but I don't, so we're just gonna move on. I'm pretty much just gonna refer to these 3 for the next section as anaerobic, and then this one as aerobic. I will use these 2 as specific examples, but I will just be referencing them as anaerobic and aerobic. So keep this in mind. Take a picture, write it down if you want, but that's what I mean when I refer to anaerobic energy. Because the next section is your different types of muscle. Smooth muscle specifically. Okay. When I'm talking about smooth muscle, I am talking, I guess, muscle in general. We talked about 3 different types. There's type 1, type 2A, and then type 2B. These are just like the categories of like, are they fast twitch, are they slow twitch, and like what kinds of energy they use. But for the purpose of this chart, I'm just going to be referencing them as type 1, type 2A, and type 2B. So our first category is the activity of ATPase. And so this first one is going to be very slow. So these are like your slow muscles. These are like not your fast twitch ones, basically your biggest ones, like your like your gastrocnemius, or maybe like your abs or your quads. Very slow muscles, like things that help you stand, things that you're going to be using for more than 1 second or 1 minute at a time. So then this one is medium, and then this one is fast. This section is pretty much just going to follow that medium all the way down, but you'll see that later. The next thing I'm going to talk about is energy, like I was saying before. So what kind of energy are these going to be using? So this one is your slow muscles are going to be using aerobic, because although aerobic sources of energy, or like your cell's metabolism, it takes a little bit longer to get energy, it's a lot more sustainable in the long run as opposed to the quicker, faster energies. So your type 2 is going to use anaerobic and aerobic, but it's going to favor anaerobic. And then this one mainly uses cyclic ATP and then creatine phosphate. And so there's what energy sources they use. So you can kind of tell, like a cyclic ATP and then the creatine phosphate, you only, like at max, you're at like 10 seconds. But then these ones are like super fast muscles. So like I'm just going to keep using the example of like your eye muscles. They're like super fast and they don't strain for very long. Like you use them a lot, but these, like you don't need an energy source that's going to last for hours if you're not going to be using that muscle for hours. And so your body's just going to make up for that, which is really convenient. All right, and the next one is fatigue. I thought this was super interesting, but I'm not going to cover it in this video, but we did cover it in class. But fatigue. So this is like a non-fatiguing muscle, if I'm thinking correct. Yes. This is a fatigue-resistant muscle. And then this is a very fatiguing muscle, because if we go back to those energy sources, if we talk about these that only last for 10 seconds, if you use them for more than 10 seconds, you're going to get fatigue. However, if you have something that is a very renewable source of energy, sorry I keep moving this board so much, then you're not going to be fatiguing as much. So if you start moving your eyes for more than 10 seconds, but you are standing for more than 10 seconds, you're going to need different energies, which makes a lot of sense. Okay, and then these ones aren't super important, so I'm not going to write them down, I'm just going to explain them. But like, the next 3 categories are the amount of mitochondria, and then the blood supply, and then the color. So these ones will be red, these ones will be white. This one has like a super high blood supply, and this one has a low blood supply, so that's why it's white. And then this one is going to have like a ton of mitochondria, and this one's just not going to have as many, because mitochondria do the aerobic respiration. Woo! Whereas glycolysis doesn't, or the anaerobic doesn't. So those are the types of energy, and then I'm going to explain the 2 types of smooth muscle movement that we do, because it's similar to the little graph, the little picture that I did earlier, but it just has a couple of different things, because smooth muscle is super interesting because it has the potential to work for so long, whereas like, and when I say so long, I mean like hours or even days on end, as opposed to you're not going to be standing for days on end unless you live a sad life. And so these 2 different types of binding between actin and myosin explains why smooth muscle is able to do that. Sorry, I also keep moving around. I'm a fidgety person. All right. I don't know why these videos make me so stressed, but they do. Anyway, the first one is called phasic. Your little phasic movement. So the pictures are going to look very similar to the first pictures. So I don't like how I did that. Again, it's just 4 steps. We have the actin, and then we have the myosin. And so I'm just going to add the basis of these just to make my life a little bit easier. Oops. So as you can see, still very similar to skeletal. However, just what you're adding and taking away during what steps are a little bit different. And so this guy, again, starts with a phosphate on it. However, during this step, if you remember before when we were talking about skeletal muscle, only calcium is added in this step. However, we're going to be adding another phosphate. We're going to be adding calcium, because when we talk about muscle, we're talking about calcium. And then we're also going to be talking about something called the myosin light chain kinase. But we do a little acronym. Is that what they're called? Acronym of I hope I'm in the picture. Myosin light chain kinase, or MLCK. And if we remember from our science background, kinase are just going to add that phosphorus, so that's how this phosphate is being added. And then, so that means that there's going to be 2 little phosphates on this guy, which is super important. So then in this step, but it can still like attach to the actin and everything, so it's not like going to change. It's not necessarily changing what it's doing, it's just changing its potential. Yeah, I like that. Okay, however, at this step, we lose, it's going to be exactly the same. We lose a phosphate, and then we lose your ADP. And then but we still have that 1 phosphate on it. So that's important. But as you can see, it's not preventing it from being in the uncocked position. So that phosphorus isn't preventing the movement anymore, whereas this one was. And so then in this guy, it just moves on to release, and then it moves on to this. So, MLC. So at this step, we have, whereas we had hydrolysis in the skeletal muscle, we're going to be adding the myosin light chain phosphatase, and then we're also going to be removing a phosphate again. And so then the ATP that we added is bound here, and then that becomes ADP, and then this is phosphorus. And so, like, the skeleton of these cycles are pretty much the same. It's just what we're adding when and why that changes. So, like, the pictures look pretty much the same, but however, this guy has the potential to override that and just keep going in this cycle. And so that's why it can just keep going. I don't know at what point, this is called phasic. I don't know at what point which muscles use phasic or tonic, because I don't think that it's like muscle specific. And so, like, whatever your smooth muscle needs, it's going to use. So if it needs phasic for some reason versus tonic, it's going to use that. I don't know what differentiates that, like if it's just like a negative feedback loop or something, but yeah, whatever. Alright. This is the last time I'm going to draw these 4 little diagrams, I promise. Alright. So this one is called, like I mentioned before, tonic, as opposed to phasic. And again, the skeleton of this is going to be exactly the same, because at the end of the day, it's all muscle and it all moves. The biggest difference between this and phasic is they use the same enzymes, it's just they use them at different steps. And so this one is still going to be using myosin light chain, this is kinase. It's still going to be adding a phosphorus. It's still going to be adding calcium. However, that just goes along with that. It's also going to be adding ATP right here. And so these cycles use a lot more ATP, but they use it a lot slower. And so, like, ATP isn't really a problem, but it's just we needed to add energy right there. And so this guy is going to have the 2 phosphates again, if we remember correctly. The ADP and the ADP. However, we are going to get rid of a phosphate, so that means we're getting rid of also our ADP, but we're also adding in our myosin light chain phosphatase right here, as opposed to over here like it was in the last cycle. So this also removes a phosphate. So in our last cycle, the phasic cycle, this one still had a phosphate on it, but right here in the tonic, we removed both phosphates. And so this guy just doesn't have anything. This guy's kind of using ATP over here. It's coming in and out, but this isn't chain. However, this guy can do something a little bit special where its return back to the cocked position can kind of like take a little bit longer. So that's why I drew the little wiggles. And this is called the latch phase. And so it just takes a little bit longer to get back to the cocked position with the phosphate and the ATP. So there's your two different types of smooth muscle. Okay, let me flip my paper. We're almost done. We're almost done. So basically the difference between your skeletal muscle and your smooth muscle is one's voluntary. Your skeletal muscle is voluntary. Smooth muscle is involuntary. Smooth muscle is controlled by the parasympathetic nervous system. So if your PNS is stimulated, like it's the dominant one, then your digestive system is basically going to be turned on. And then if it's not, your digestive system is not on. Smooth muscle is pretty much just found in your like your organ cavities, not really anywhere else. Yeah. Oh, skeletal muscle is also striated, and smooth muscle is not. Those are the differences. Last thing I wanted to talk about is your muscle sensory receptors. And so, this is, I think I say and so too much. However, this is talking about reflexes. So your muscles, like your skeletal muscles, are voluntary. That's not a lie. Did not lie to you. But you're not necessarily always thinking about it. Like you don't have to actively be thinking about standing. You just do it. But like as a child, you have to learn how to stand. But like you're always making minor adjustments. You're always moving around. You're always trying to keep your balance, stuff like that. And that's stuff that you don't necessarily control. So that's why we have reflexes. But a really good example of a reflex is the one you get like at the doctor's office. And so you basically have your little muscle, your little leg here. Here's your little foot. Leg. We have your quads up here, and then we have your hamstrings down here. So your quad muscles have your little patellar tendon that wraps around your knee. It's at that bent position. What you do is you hit that with a hammer gently, which stimulates your stretch response in your quads. And so I'll draw it out in a second, but basically it excites this muscle, so this muscle contracts, and then it inhibits this muscle, so it relaxes, and so then your foot can go up and out. So then you just, like kick a little. So I'm gonna show how that actually works. So this is your spinal cord. So we have your stimulus coming over here, coming in here. It's going to tell two different motor neurons. I think they're motor neurons, right? Yeah. You're going to have an agonist and an antagonist neuron. That's going to come out here. They're going to synapse with these two guys. This one and this one. So your agonist is what's going to be working with the stimulus. And so that would be, this guy would come all the way up to your quads and stimulate them to contract. So that's why it's called agonist, is because it's going to be working with you. And then your antagonist is going to be coming up and going to your hamstrings, which is going to tell your hamstrings to not contract. And that's what allows your foot to move forward. If we didn't inhibit this muscle, then your leg would just like tense up and like you wouldn't move. But like if we inhibited this one and then excited this one, then your leg would go this way. Oops. This way, because you're contracting this one and relaxing that one. So this happens all over your body. So you could say this is your bicep and this is your tricep. And when you when you excite this one, then your arm's gonna go like, woo! But then in order to do that, your tricep has to be inhibited and told to not move. that's your stretch response. Yeah, that's pretty much all about muscles. I hope I answered every question. This is a super cool unit. Thanks for listening.